Okay. Well, hello everyone, and welcome to the DHCon seminar series. I feel very special today that I get to introduce Dr. Marcus Call, a professor in the Division of Biomedical Sciences at the UC Riverside School of Medicine. So Dr. Call's graduate training is in pharmacy and pharmacology, and then he came to the United States in 1997 for postdoc work at Brigham Women's Hospital at Harvard Medical School. And soon after, he was recruited to the Burnham Institute in La Jolla, where he also had an appointment in the Department of Psychiatry at UCSD. So I'm sure many of you are familiar with at least some aspect of his very large body of work. He's widely recognized in the NeuroHIV and neuroimmune fields. Not that numbers are everything, but he has an H index of 40, and his work has been cited over 8,600 times. His lab investigates the mechanisms of chronic infection and persistence in both HIV and now COVID as well, with a focus on immune signaling that can tip the brain toward injury or protection. And he also investigates the influence of drugs of abuse on neural plasticity and host pathogen interactions. I found an article he was quoted on about the interactions between HIV antiretrovirals and methamphetamines, where he described the biology as an unpredictable storm, which is such a perfect metaphor for this kind of work because it has messy inputs and nonlinear biology, but also very real stakes. So, if you're not familiar with all of his papers, Dr. Call is also a regular attendee of this seminar series, and it's hard not to notice that he consistently contributes and asks insightful, constructive questions to help move science forward, but also to help build a sense of community, where we know that we should all feel comfortable to interact and ask questions. So today we get to hear him speak again, and we get to be the ones asking him questions. His talk will be on ephrin signaling in microglia and implications for NeuroHIV. So thank you for joining us, Dr. Call, and the Zoom floor is yours.
Yeah, thank you, Dr. Gasnick. Thank you very much for this very nice introduction. And, no, I'm really happy to be here. I really enjoy this seminar series. I think it's great to have that, in a great forum to discuss science. And so I'm feel really lucky to, and an honor to have a chance to present you with some of the work that we've been doing in there. And may have to introduce today, a little bit EphB2-ephrin-B1 signaling in microglia and implication in neurons, as the title says. Let me see. How do I? Sorry, my computer is a little bit slow. Oh, okay. So let me start with just introducing a little bit the people who have contributed to this. Can you see my cursor?
Yes.
Sorry, yes.
Yeah.
Okay. Okay. Great. Okay. So what I'm presenting today is really work of primarily, done by one of the graduate students in the lab who recently graduated and now decided to go into industry, together with who is now actually his wife, Samantha. So Jeff and Samantha, basically put their heads together as graduate students, and and we're in two different labs, and we're starting to discuss science. And then one day Jeff came to me and said, we've looked at the data, and so we came upon this efference, and it just happens to be that Samantha's supervisor, Dr. Renee Athel, who is another collaborator in this story, actually works on efference. So efferents were pretty new to us at the beginning of this, but not totally unfamiliar, because we had noticed that they came up in gene expression studies that we have done, and then in the bioinformatics analysis. So we didn't see too much of a different regulation of them, of these components, but they seem to have a prominent role in some of the gene networks. Now, however, all the work I'm talking about today is really embedded in a work that a lot of other people have contributed to over time. And so we are really thankful for have all these collaborators. So the ones shown in red here are involved in the study or have contributed to the study, including Ben Gelman through the NNTC, and Amanda Roberts, who is our collaborator on all things animal models and behavior. And of course, finally, I want to acknowledge the NIH and the funding sources. Without those, we really couldn't do a lot of this work. Okay, so let's get into some of the, for those, if there are any, or if there is anyone who is not so familiar with this, just, for those who know all this, please forgive me if I just, tell you something that you know anyway. But for those who are maybe newer participants, just briefly something about HIV, neuro-HIV, neurocognitive impairment, and this term HIV-associated neurocognitive disorder. So the prevalence in HIV, even at um, 2025, is about worldwide 40.8 million people. In the US, it seems to be fairly stable, I think, over years. So some of you probably know this better than me, about 1.1 to 1.2 million. And it looks like the latest numbers from CDC and HIV.gov say that 87% of HIV-positive individuals actually know about their infection. So we have still about 13% of people who might not even know. And of those, 67% seem to be virally suppressed. And again, some of you who work actually with patients, maybe correct me if these numbers are, if you know about different numbers. It looks like that even though with very good treatment, about 30 to 60% of HIV patients, depending on the cohort, experience still neurological complications, including neurocognitive impairment. And summarily, we often still refer to this as neuro-AIDS. In the earlier times now, for many years now, we talk more about neuro-HIV, since AIDS has become simply less frequent. So, and the HIV associated neurocognitive disorders still also seem to appear eventually in about 50% of HIV patients, right? So the categories, many of you are very familiar with for hand are asymptomatic neurocognitive impairment, mild, and then dementia is the most severe component. Now, a colleague from Johns Hopkins, almost now 10 years ago, right? They published what is really, I think, striking, that in between the pre-CART era and the CART era, which works really, really well to suppress viral propagation, we still, however, have about 50% of people who have some level of impairment. Dementia is much less, but mild neurocognitive impairment and asymptomatic neurocognitive impairment still have increased. So you wonder why these numbers don't really go down when we still have only about half of the people who actually show no impairment. So, and even though a lot fewer people now come to autopsy, luckily, because people live longer with HIV, those who come or are analyzed seem to show still neuropathological abnormalities in about 80% of the cases. So again, this is what what is still occurring, and even so, these major neuropathological signs, which include like loss of dendrites, synapses, and gliosis, like microglial activation, astrocyte activation, astrocytosis, also still do occur apparently, despite suppression of the viruses. So I'm now showing you something that's also kind of older, but it still seems to hold true to this day. HIV infection, of course, always occurs in the periphery, but the virus makes the way into the brain apparently very quickly, where it's passed on to local microglia, which then can pretty quickly, start not only to produce virus. Overall, it seems like microglial cells produce relatively little virus, but they become activated and they release factors that can pretty directly affect neurons. This includes the viral proteins themselves, but also neurotoxins, and they also produce as a part of the immune response, an activation cytokines and in particular chemokines. Now, those then also have a role in the activation of astrocytes. Now we know, and this is, there's still, I think, debate about this, that the virus sometimes makes their way into astrocytes, even so astrocytes don't seem to be, good at producing, so doesn't seem to be a productive infection. However, astrocytes in this scenario also change dramatically, reducing the amount of growth factors they produce. They might also they play an important role in glutamate or in general in neurotransmitter homeostasis. So they may change that. And in extreme cases, it's been reported that it might even possible that they start to release glutamate. What they also produce sometimes is these oxidative species like NO, which also then can hit neurons. So you get basically a situation where the microglia as well as the astrocytes start to compromise neuronal function. In neurons, we still see that there is problems with calcium homeostasis apparently. The glutamate, glutamate release or response to glutamate may go up, which I always, since I entered the field, found a little bit, yeah, kind of surprising because a lot of data indicate that people who actually have cognitive impairment have not too much; they have rather less glutamate. But this is not necessarily exclusive. So this situation we can also discuss the details. It's very interesting to see. But again, these mechanisms still apply after so many years where we have good treatment for HIV. Now, the interesting thing about microglia is that even if only a few of these cells are infected, they don't need to be infected actually to become a problem because they also just recognize some viral proteins, which is sufficient actually to get them to produce neurotoxins. So this setting still seems to, a lot of the data that we find still fit with this kind of model. So we are now basically trying to better understand what are exactly the molecular and cellular mechanisms in this setting. So CRT, again, very important in blocking HIV infection. And what I will be talking about today is basically something where we will see where HIV might be an initiating factor, but even if we then remove HIV from the picture, some of the processes that have been initiated might actually go on independently of this and can still cause trouble potentially. So, and this is where we now have to introduce ephrin and ephrin signaling. So, ephrin and ephrin signaling is in principle well known, not so much studied in the context of HIV. But there are some interesting characteristics of the system because we have here basically what is called these ligands, which are actually in most cases membrane bound. They either GPI anchored or really membrane proteins, and they can also be released. So they're soluble functional. So these are basically considered the ligands, and then we have the receptors, the FAB. So this is a whole family of proteins. It's not only these are just some of them. And the functionally interesting part is that these molecules actually mediate bidirectional signaling. So it's generally first referred to as reverse signaling when actually the ligand signal through interaction with the receptor signals back into the ligand producing cell. And we know about that they signal in terms of adhesion, cell migration, proliferations, and people have looked a lot. Oops, sorry. Some of the pathways in there. So, and then if the receptor is activated, we also get forward signaling, which also influences adhesion, synaptic plasticity, neural outgrowth, and proliferation. So in the nervous system, we know a lot of this. In fact, best known functions of the system, besides the bidirectional signal and cell-cell contact, is synaptic plasticity, cell migration, cell adhesion, and repulsion. So they play a role in both. And this has been studied in particular in the context of neural development in cancer. Now, more recently, it has been recognized that these factors also seem to play a function in inflammation. So there we are probably still in for some interesting discoveries because this part has been relatively understudied a little bit. So how did we get back into this? So let me again back up a little bit. Initially, we got then into this, as I mentioned before, because we have been doing a lot of studies with a transgenic mouse model for HIV-associated injury. And this is the HIV GBM20 transgenic mouse that express it in astrocytes under GFAP promoter, the Leonard Mucki generated mouse, and 30 years ago. And but it still has a very reliable phenotype, and that you can see loss of dendrites, loss of synapses, activated microglia, astrocytosis. You see compromised neurogenesis in these mice. You see behavioral changes. So there are also electrophysiological alterations. They are not so, we can't, a lot of these neuropathological things fit very well with what has been observed in humans. And electrophysiological alteration is a little bit more difficult to compare directly. And then there are also endocrine system alterations. The same is true here. But finally, what we found, now it's also quite some years ago, that there's a significant overlap in differential gene expression with human HIV brains in these mouse brains. And finally, we saw in this context that EphB2 and the ligands FNB, the gene name is FNB for ephrin-B, are components of the top functional neural networks. So, okay, so we saw Okay, after Jeff came and talked about this, let's take a look at this. So we let's look a little bit what is known about ephrin-B2 in the periphery and driving inflammation. So there have been some studies. This came out in 2018 that showed that ephrin-B2 was prominently expressed in liver inflammation and fibrosis in a mouse model of tetrachloride carbon, which is a very pronounced liver toxin. But the same study overall observed that actually ephrin-B2 was upregulated in human liver cirrhosis models. So, and then they looked at the whole panel of inflammatory or or inflammation-related factors, and they found that there was significant regulation of this. And they also did some of the studies in ephrin-B2 knockout animals. They could see that a lot of these factors were significantly downregulated. So, and overall, they found that ephrin-B2 seemed to promote inflammation and fibrosis, and if they blocked it or basically knocked it out, a lot of these inflammatory responses and the fibrosis were actually avoidable. Now, we went back then to the NNTC cohort, and this is where Dr. Ben Gelman was really important, who had helped us to obtain samples here from the NNTC that we have been using in a couple of studies. This is most based really on mRNA expression here, and was mostly quantitative reverse transcription PCR. This cohort that he has characterized also in other publications includes non-infected controls as well as a group of HIV patients. Most of these have been hard treated, but they are not virally suppressed for the most part in here. Okay, from those samples, when Jeff started to look at it, initially he said, oh, okay, if we just look at non-infectious versus HIV, there's not really much, there's maybe a little bit higher, but it was not significant. However, once he started to separate the samples out by brain samples with brain pathology and without brain pathology, you could see that in the samples without brain pathology, everything was basically normal, but expression was up in those that showed brain pathology. Okay, we said, okay, so maybe there is something to that. So, and then as we looked more through the data, it became clear that ephrin-B2 expression actually did correlate with mRNA expression in the CNS, and also with the DNA in the cortex. So that was okay. So there might be another link. So it's just correlation, right? We don't know any causal link, but it indicated that maybe ephrins do play a role in this context. And then, as Jeff went further into this, he also found that ephrin was basically inversely to abstract executive domain scores, so had some behavioral or some correlation with behavioral outcomes as well as verbal fluency here. Okay, so my correlations may not look really dramatic, or the p-value is not very high, but it indicated that that there might be something going on in this case. Now, then we were not the only ones, as it turns out. Looking at this, there was a study by Ufarov and I used samples from the Manhattan Green Bank and looked particularly the anterior cingulate and also the caudate, and they found also actually something here. So they didn't really differentiate between neuropathology or not, that what we did in our study, but they found that ephrin-B2 was more upregulated actually in those who had showed cognitive impairment, and it also correlated with worse motor T-scores as well as obstructive executive functional T-scores. So now there was basically some independent, indication that the ephrins might play a role, or could be contributing to some of the effects in the brains of people living with HIV. Again, so far all correlation, it doesn't mean that that there is any functional role into this. And this was really what we were wondering about. So the question was, does this axis really play a mechanistic role in brain injury potentially? Or by injury by HIV infection itself, or perhaps the host response, right? Because the efferents are part of the host response. So we also were wondering, so where exactly are the locations? So the studies so far didn't really indicate it, and then really get down to the question of what does it really do. So we went back to the mouse model, right? And so we went back to some of the tissues that we had actually banked from earlier studies. And I don't know, you may remember, some of you may remember that a few years back, we did a study where we, tried to figure out if interferon beta could actually be protective. And we found that intranasal interferon beta treatment of the mice could actually prevent toxicity relatively. We were surprised it worked really well and at a relatively short so we went back to some of these samples, and what it turns out that interferon beta treatment as well as the GB120, the presence of the GB120 seemed to increase the expression of ephrin-B1 in the CA1 region, you can see, but also in particular in microglia. So again, not very dramatic effect, and I'm showing you here the data that basically has all the data points here, because it shows a little bit more what the distribution is. So the effect is not, I mean, it reaches, if you look at all the data points, significance for the total CA. In the microglia, the interferon beta does almost, is the trend, but with the transgenic, so you have this upregulation. Now, our collaborator, Dr. Ethel, was really excited when she saw this because she said, oh, we didn't really know that interference might be regulating the system. So that, okay, to us, it was also completely new, but we were a little bit less, biased in any way because we really didn't know what to expect at this point. Meanwhile, if you go look in the literature, so people have been looking at ephrin in the context of the innate immune response, of course, but there's no understanding that the interferon system directly regulates the system. Okay, so that was another interesting question coming up, and this is why I'm showing here that a little bit just a simplified scheme of what the interferon response in HIV infection might look like, what we know, the REI system, and then TREF2 signaling, and finally we get IF3 and IF7 activated in order to get some of the type 1 interferons themselves expressed, but also some of the, then in response to the interference that can act autocrine or paracrine, and then so if now 1, 2, and then basically trigger also some of the expression of the interferon response proteins, or the interferon stimulated genes are they often referred to. So when we looked then at some samples, because we had also done studies with the interferon beta knockout, so what we saw then, and again, these effects are not very dramatic, but for the ligand, we could see there was a trend that in the interferon beta knockout, this is without GB120, that there was a reduction in the expression and the homeostatic or baseline expression, and it was really significant actually for EphB2. So this was another indication that the interferon system and the ephrin system might actually be interacting, and that the interferon system might actually play a regulatory role in the system. So, and then looking more into what happens in the interferon beta knockout, of course, maybe less surprising is that it downregulates also the expression of IF7. Initially, we thought, let's just look at some of this as a control, but we can really see that the GB120 actually is driving in these brains IF7 expression. But if we knock out interferon beta, it's basically back to baseline. And this is true in the cortex and also in the hippocampus here. So, okay, so this is kind of what you would expect, right? So, but it also pointed to raise some interesting question about what IRF7, because IRF7 is also known to be regulating, some aspects of HIV infection. And is a part of the host response. So Jeff and Samantha and we were just discussing this. Look, let's see what if IIF7 has any more role in this, right? So first of all, going back to the NNTC samples, it's not surprising perhaps that IF7 is upregulated with the HIV infection. Is kind of what you would expect. But again, similar to what happened with EphB2, it's actually in people who have no brain pathology, it's actually not significantly upregulated, but it is significantly upregulated in the people who have brain pathology. So this is a surprising similarity with what we saw with the ephrin-B2 in this case, right? And then looking at IF7 and ephrin-B1, the ligand as well as the receptor, the FB2, we again saw that there's a pretty good correlation in the expression for these two factors. Again, it's only correlations, but it raised, question, do they have anything to do with each other in terms of function, right? So, so as we started then to move a little bit more into IF7, and we went back into a microglia model. We used here first the HMC3 cells and looked a little bit what happens here. So if you expose these cells to interferon beta, right, you get IF7 upregulated that you would expect, and the siRNA for IF7 shows that you can completely, prevent this basically. So actually the baseline level really go down with the siRNA. So the siRNA works really well, right? So what consequences has it otherwise? Now, if we look at FB2, it's actually surprising because if we knock down IF7, FB1 is even more upregulated, right? And yeah, in response to interferon beta. That was interesting. So it looks like IF7 actually puts perhaps the brakes on FB2 expression. The same happens with EphB2-1, right? Another interferon stimulated gene. IF7, reducing IF7 basically takes off the brakes. And then most interestingly, interferon beta itself induces more interferon beta in these microglial cells. Again, if we knock down IF7, we basically take the brakes off on this. So IF7, basically turns out or looks like a negative feedback regulator for some of these pathways related to interferon beta signaling as well as ephrin signaling, or ephrin expression at least. Yeah. So again, our collaborator, Dr. Ethel, was really excited about this because for the ephrin field, this was apparently completely new, right? So at this point, where are we? Interferon beta and HIV GB120 expression seem to drive, a moderate upregulation in the model of EphB1 as well as FB2. If we ablate, or knock out interferon beta, that reduces the EphB1, FB2 RNA expression levels. EphB1 on microglia seems to be regulated by HIV GB120 and interferon beta. This is what we basically concluded from the mouse model. Again, even if it's relatively moderate. And microglial interferon beta, EphB1, FB is mediated by type 1 interferon master regulator IRF7, basically. So in particular, in terms of it looks like a negative feedback loop, right? And there. Okay, so far so good. But what does it mean, right? So what really does this axis now do? Now they are regular and we have some regulation that links it to the interferon. We know that interferons themselves have a pretty pronounced effect on what is happening in HIV infection. But now it was, so what is really going on functionally here, or is anything going on really functionally with EphB1 in terms of inflammation or neurotoxicity, considering what people had been observing in the periphery. So we went back then and used here then a system that our collaborator Esther had evolved, and that is commonly used apparently in the ephrin field, that you basically pre-cluster FB2 recombinant protein with a specific antibody. This is fusion protein of FB2 with Fc part. So with an antibody against the Fc part, you can basically aggregate it. So you basically produce an aggregate that is actually only temporarily stable. So there is a certain time limit to this. You basically prepare these aggregates and then you need to put them onto the cells in order to get the stimulation, and they're stable for maybe up to 2 hours, and then they basically become inactive, these aggregates. But it is an established way to basically basically trigger or perform FB2 dependent stimulation of cells. So we applied this here to the microglia, and then we basically let the cells go for 24 hours. And then connected the cells as well as swelling, but here in particular, looked first at the RNA and did some RNA sequencing just to see how would the cell respond. And to make a long story short, so this is just showing some of the data. So you get what you can see here. There is really significant upregulation of several of the innate immune response genes, including actually some of the interferon response genes, but they're also like complement components, interleukins, IL-6. So here is basically showing what we see. IL-6 is upregulated significantly, and CD68 in the cells is also upregulated as a marker also of activation of these cells. So on the right side, we see a little bit like a heat map of this, how these were three experiments here, the vehicle treatments and then the EphB2 treatments were there. And yeah, there is some variability between the batches, but you can see it's pretty consistent how they respond. So it basically indicated that EphB2 activation can drive, in some components of an antiviral, but at the same time also an inflammatory response in microglia. In there. So if you then analyze this a little bit more, use some of the bioinformatics, what you can see specifically. So clearly, even, and interesting, this is just the EphB2. It does, trigger some components of the interferon beta response. So there seems to be more of a link, perhaps unexpected link between the ephrin signaling and what interferon does. Negative regulation of biogenome. So this would all fit with this. And so there seems to be a strong aspect of antiviral immune response, but there is also clearly like cytokine-mediated signaling pathways, right? So that point to some aspect of really inflammatory activation. So it's a little bit a network here that we used, that IPA gives one of the top networks here. Again, we do not yet know what all of these factors, exactly contributed, but you can see that some of these factors significantly upregulated. But there's also certain factors that are inhibited. So what this whole, expression pattern basically suggests is that we're looking here at something that is, speaks towards the balance of antiviral activity versus inflammatory response, right? In there. So in order then to further see how much of this might be actually really driven by EphB1, we went on, or Jeff went on really to try out the knockout of EphB1 and then perform the EphB2 treatment in the microglial cells. So again, was using some siRNA to knock down EphB1 and then stimulate the knockdown cells as well as the wild type or controlled cells with ephrin as we saw before. And again, we looked for some of the sequences as well, multiplexes, and eventually also we wanted to look, does it have any role in neurotoxicity? So let's look at this. When we just let's look a little bit at how effective is actually the siRNA knockdown. And it leads, even with the best siRNA, we get about a 50. So we don't get a complete knockdown in there, but we get a 50% knockdown. And this is reflected also at the protein level, really, that there is. So we couldn't find any siRNA that would really completely knock it down. But okay, so we had a 50% knockdown, and we would think that perhaps this is sufficient to see some biological effect as well in there. So going back then, and this is not gene expression here, this is just looking at some of the multiplex assay, the secretome in terms of the proteins that are affected by this. So you have here the vehicle, the ephrin, or the control, basically EphB2 stimulation, and this, and then compared to what we see in the siRNA knockdown of EphB1. So we can see that there is a gradual difference. It's not a complete prevention of induction of some of these immune factors or interferon response factors. We can see here, or factors that have, some of them have linked to neural injury too, like the MMPs, for example. And here's just some examples. So the IP10, for example, you get a knockdown, not a complete, down to baseline, but this is a significant reduction of the expression of this. GM-CSF was, one of the, where we saw some of the most pronounced effects, eotaxin as well as MMP9. So some of the factors that are really downregulated. So again, if you knock down EphB1 and stimulate EphB2, so it's clearly there seems to be a role for EphB2, EphB1 signaling in driving some of these factors in the expression here. So if we now look at this, does it have any relevance, with antiviral and effects and inflammation on actual HIV infection? So Jeff then, and this is still work in progress, but in one of the first or last of experiments that he did during his PhD work, and one of the first experiments leading into this field, we went back to iPSC-derived microglia here, and those are the ones from Fujifilm CDI. We put them in culture, you infect them with HIV, and this experiment went over 12 days. So during this time, we measured P24 just to make sure that they have and the infection actually works quite well. This actually surprisingly well in these microglial cells. So this is something for a different talk, HIV infection of these iPSC-induced microglia. So, but we collected, supernatants and over time, and then at the end of the 12 days, basically the cells first, in this case, really to look at what is there anything happening with HIV. So, and what we can see here, so it's just looking at two of the genes here. This was while GP120 and TAT, where you look at RT-PCR, and you can see ephrin-B2 expression. So this is not the perfect way actually to show that, but when Jeff put this together, but you can see there's a significant drop in GP120 message as well as in the TAT message after stimulation with EphB2. It's not that it goes completely down, but there is a reduction of this. So that basically fits with the idea that the ephrin-B2 response might be part of or component of the antiviral response that we can see, right? So again, this speaks to the balance. The system or the brain maybe tries to do something or tries to limit combined infection, and one of the components might be actually the ephrin signaling that is contributing to this. So, but now getting to the other side, because neural injury, is there anything going on? Is there any role for this actually in the neural injury? So here we also started then to work with iPSC-derived neuron and astrocyte co-cultures. Again, in this case, they were also used. We used the cells that are basically available from Fujifilm CDI. And then, so we triggered again microglia with ephrin-B1 siRNA, and then or control RNA, and then stimulated them with ephrin-B2, collected the supernatants, and then transferred those supernatants onto these iPSC-derived neurons and astrocyte co-cultures. So let's see what happens then. So what we can actually see here is that, oops, wrong direction, that in fact, ephrin-B2 stimulation does cause some neurotoxicity, these cells to produce some neurotoxicity, right? So again, we haven't played around with some of the concentrations. This was just something that seemed to work pretty well, but it indicates that with this antiviral, limited antiviral response, we might already see some basically undesired effect of neuronal injury in this. And that is basically becomes ameliorated, namely reduced, if we knock down ephrin-B1. So it looks really like that ephrin-B1, EphB2 signaling might contribute to antiviral response, but at the expense of also be able to causing some neurotoxicity. So then, to wrap this up, basically what we think is happening here, that ephrin-B2 can contribute to HIV-induced neuronal injury because simply HIV infection triggers this. But as you saw in the model system, this is basically we no longer need to have HIV in there. Basically, the ephrin-B2 signaling alone would be sufficient to trigger the cells to become, to keep up some antiviral response, but also actually to get some neurotoxicity here. So I didn't show you this data. ephrin-B2 actually also induces NF-κB signaling, which may feed into the inflammatory responses. But it also has this this antiviral response again at the expense an antiviral response at the expense of an inflammatory and neurotoxic response. So if we were then to basically reduce ephrin-2 signaling, for example, by No, in this case, the siRNA for EphB2, for EphB2-ephrin-B1 in microglia, then we might be able to we would probably reduce the antiviral response of the microglia themselves, but we would also perhaps be able to really reduce some of the neurotoxicity. The question really then comes is there a way to, think not in terms of a switch of on and off, but rather to modulate it, like to tune the response of the cells to the way to reduce it just enough to prevent neurotoxicity. So the jury is still out on this. But the other important point is here that this system seems to be, able to work, once and then the inflammatory response or the expression of these factors are basically initiated by HIV. We can take away HIV basically. So we can, if you have viral suppression, but if you might no longer have HIV that pushes or triggers the system. But if the system is already activated and has basically become a self-maintaining inflammatory response, you might still see some of the problems that we were just looking at here. Okay, and I think this basically ends the story, and I will be happy to answer or discuss some questions. Thank you.
Thank you. We don't have any questions in the chat yet, but if people want to put them in the chat, I can read them out if you're more comfortable that way, or feel free to unmute yourselves and ask.
I hope I wasn't going too fast.
Hello. Hello.
Hi, Peter.
Hey, Marcus, how are you?
Good, how are you?
That was great. Thank you. I wanted to ask, so you were talking earlier at the beginning, you talked about how GP120 and interferon beta both act on FB1, FB2 and FB1, right?
The expression, they seem
Yeah,
to
the
drive out the
expression.
expression.
But I was wondering, do you think that GP120 acts on the cell to drive changes in interferon beta that then act on FB1? Like, are they sequential? Do they just happen to use the same pathway? If you do them together, is the pathway enhanced or is there overlap? Like, how do the two interact? Because it seems unlikely that they both do exactly the same thing but don't interact with each other.
No, it's a really good question. And so we don't know, we cannot totally exclude that the GP120 protein itself, could drive this. But what we actually suspect is that in the transgenic mouse, because in the transgenic mouse we see an interferon response, interferon beta response, and we see a signature of an interferon response. So we suspect that because the transgenic model does actually express, the original viral sequence, RNA sequence, and we suspect that this might be driving an interferon response, and that might then be driving the ephrin
Because
responses
you've probably done it with a non-HIV associated interferon driver as a control, right? Do you see the same effect, right? I'm wondering is this specific to GP120 or is it more likely just a general aspect of response to the body saying, oh, there's virus here?
Yes, and it might, I don't think this is necessarily specific to HIV. I think this can happen with other viruses as well. In fact, so there's some literature out there suggesting that EBV, does trigger something similar in B cells, right? And the B cell setting and endothelial cells as well. So no, I think we might here be looking at something that is just part of the host response to viruses. And it just happens to be that in this case, the HIV is the virus, and it's also in the brain, that it just, and, you could imagine that, and we were wondering too, has anyone looked in the context of herpes viruses or, or some of the other viruses to make it into the brain? And honestly, we don't know.
Okay, cool. Thanks, Marcus.
Sure.
Hi, Marcus. I'll go ahead and jump on.
Oh, okay. Hi,
Much.
Amanda.
Hi, good. Yeah, enjoyed a little bit late, but glad I didn't miss it. Very, very interesting work in this role of the ephrin B, the ligand, and the receptor. And so in your co-culture systems, so you've looked at neurocytotoxicity. So have you also looked at, what happens to astrocytes?
Yes, good. Very interesting question. So this is actually something that is still, work ongoing. So, and because initially when we talked to our collaborator, Irina Asmus, so she had been looking at this in the context of traumatic brain injury. And in that context, actually, it looks like that the FB2 is primarily expressed an increase in expression in astrocytes. So this is kind of why we see it in the stainings in the mouse also. It also overlaps with astrocytes. It seems to be interesting. It actually FB2 is also expressed in microglia themselves, right? So there seems to be a component of paracrine as well as probably some autocrine signaling in there, depending actually on how exactly the cells are stimulated. So it's a really good question. So at this point, we think that it most likely we suspect that because we were a little bit limited because the FB2 antibodies we haven't really found one that is that good, to do some of the staining, and but the ephrin-B1 worked really well. So this is where we can see the ligand, but our collaborator now has a knockout for astrocyte-specific knockout. So this is something we would like to see, try. But we're moving away a little bit from the transgenic models. So this is why the mouse work is basically, basically a little bit on hold right now because with everything moving more into humanized mouse models. But the transgenic models really have still this, have some advantages in that you can just crossbreed them with some of the other genetic knockouts, right? And then so this is something we would like to do. So
Great. Thank you so much.
sure.
Yeah. There is a question for you in the chat that I'll read out
Oh,
from
okay.
Dr. Tekwani. He asks
Yeah.
if the FB2 treated microglia release neurotoxic factors, if you know what they are in the conditioned media, if you have an idea of what might be driving the neuronal death, and if there's any effect after clearing HIV with antiretroviral treatment, and how long the effect might remain.
Oh, okay. Good. All good question. Thanks. So we have not specifically nailed down which of the factors. So there is a whole panel of factors that are released in response to FB2 things. So this is what I showed in the secretome. So, and there are several candidates there that have been implicated in neurotoxicity. Anywhere from IP10 to or CXCL10 to MMP9. So it's we think our hypothesis it's not one single factor. It might be a whole panel of factors that might actually act on the neurons then. But we haven't, this is a really good question, and it would be interesting us to drill down a little bit more on is there a factor that is particularly important in this? At the same time, we see also induction of some of these. So we know that, for example, from earlier work that CCL4 and even CCL5 can be neuroprotective factors. It depends on how much of them is expressed. And CCL5 is an interesting factor because whether or not it is protective depends actually on that you have enough and that it's early enough induced. We learned this from the interferon response because what we find with the interferon beta, the protective effect of interferon beta strongly seems to depend on CCL4, and this was initially a little bit surprising because we know that CCL5 is also considered to be a good antiviral factor, but it simply needs longer to be induced. CCL4, CCL3 come up very quickly in interferon response, whereas CCL5 just needs a little bit longer to be induced. But if it reaches high enough levels, eventually, then it can be neuroprotective as well as antiviral, right, in there. So and coming back, so then the other factors that are induced, we would, in the past, you would try to, fish out some of these factors, just reduced to see, how many factors do you need removed in order to prevent toxicity, or is there some of the receptors for some of the factors that you can block, and then that might indicate, that there is that they play a critical role in the toxicity. But again, in the broader case, we think it's not only one factor, that we think it's a whole panel of factors that these cells release in response to FB2 signaling, and that the aggregate of these factors might be the problem for the neurons.
Yeah. There's a hand raised. Do you want to
Oh, yeah, Eliseo.
yourself?
Hi, Theo.
Hey, hi, Marcos. Good to see you. Oh, sorry, cover my camera. Then good to see you, Marcos. Then really interesting presentation. Then the ephrin receptor and the ligand, similar to the question that Amanda did, then they are essential also for synaptic formation, axon guidance, and stuff like that. Do you have
Yes.
any data in synaptic strength and stuff like that related to that?
Really good question. And
Also for stem cells, because in the animal, probably one of the places that they're more expressed is where in the subventricular zone and stuff like that, that's
yeah.
not been proven that is essential for memory, but looks so exciting that it's like, wow.
You have it's a really good point. And then so we know and we can see in the mouse, where it's up, we can see that there's a loss of presynaptic terminals, right? So and this is something that has been observed in a postmortem specimen of humans as well, right?
Yeah.
Could this be driven by absolutely? I think after this data, yes, you would wonder, right? And
Yeah, because it's so clean in that sense that you expect that you want to see synaptic and axon shrinking or growing and stuff like that, depending on the expression of GP120, and that can give you a really nice mechanism. It was beautiful.
Yes, no, absolutely. In fact, Yufarov and colleagues, then that looked at also some of the NT-subs also were proposing it. We're asking a kind of a similar question, right? And I think it's again, it comes down to that we know that the system is really important for for neuronal guidance, for synapse formation, maintaining, and probably for plasticity, neuronal plasticity as well. So this is why it is. I mean, here in terms of toxicity, we think, okay, yeah, no, maybe we could, if we could target something specifically to the microglia, if we are worried that they are, causing, neurotoxicity. But yes, you would always be concerned, if we interfere with the system, how does it affect actually neuroplasticity, right? But if you think about this, so what I think emerges, from your work and work of, and all of us in the field is that we see these changes in inflammatory factors and antiviral factors, right? And that we learn more that a lot of the cytokines and innate immune factors, like anything from the complement components to these, do not only have, effects in terms of the immune system, maintaining, but they also have some other physiological effects, right? So,
for the
and
point of the development, Marcus, I think it is amazing the data, and this is so important
thank you.
for development migration that is beautiful.
Yeah, I think the question that we end up with is, is again, this is why I was saying, we're looking here not on an on and off system. It's really probably about modulating it and tuning it, just to the right level, right? And coming back to this kind of having a balance between having, enough viral control versus, not having too much inflammation and hopefully no neurotoxicity, right? Ideally. And I think now it would be, I mean, since we see, our, like data and the data from Yuvarov and the NC would indicate that actually there is something that can, if EphB2 in particular now is not upregulated, right? That at least correlates, with less or no impairment, right? Functional impairment. So so maybe this is if there would be a way to return things to baseline levels, right? Not switching them off completely, but returning them to baseline. Maybe this is something, but the question is how to achieve that. Right?
Yeah, pretty good.
Because it looks like the antivirals, even if they're good in suppression, may not be able to do that, right?
Yeah. Good to see you, man.
Thank you. Sure.
I'm glad you got a lot of questions. I have one too. Could
Yes,
you add an Ephase or an MEA sort of experimental paradigm to your same condition media type of experimental setup?
Yes, we would love to do this. In fact, we are talking to a colleague who just recently joined UCR, who is we're thinking about some MEA experiments, with this microelectrode arrays. We would really love to do this right now. We haven't had the chance to do that, but you're absolutely right. I think this would be really helpful in order to get to see how it affects actually just neural signal. Because we also we know now. I mean, many of us have seen that a lot of things happen in neurons and functional changes occur before we lose the neurons or before they or we lose or the structural integrity of the neurons is affected, right? So I absolutely agree. I mean, electrophysiology would be a really, really nice way to look at this. Oh, there was,
I have terrible luck and my internet went out just as you were answering the question. But I look forward
could you
to
hear me?
listening to the recording.
Oh, no. No, I was just saying, you're absolutely right. I mean, electrophysiology, I think, would be really interesting now to go. And I have a colleague who has just recently joined and who is interested also in doing this kind of, in a different context. She looks at neuroinflammation completely independent of HIV infection. But we're basically thinking about the same type of experiment. And you're absolutely right. I mean, electrophysiology would probably be really nice to see. In particular, if
Because you could do it on your cell culture, but also slices in the mouse model to look at
right.
LP or something. If you end up going down this line, I would love to help with analysis with all of my
Oh,
extra
okay.
bandwidth. But we'll
Thank you.
see.
Yeah.
We'll see.
We may, no, I appreciate that because we really may need that. No, thank you.
Well, thank you for your talk today. I think everyone was very interested in it. I see Carolyn
Thank you.
came off uh came on camera. Were you going to make an announcement?
Yes. So next week's talk is cerebral blood flow and blood-brain barrier permeability in chronically HIV-infected mice on ART, and it's with Amanda Brown from Johns Hopkins School of Medicine and JH Khan.
All right. Thank you again so much, Dr. Call. We will
Thank
see you
you.
every
Thank
week
you.
from now.
You next week. Thank you.
Bye.
Bye, Marcus. Great job.
Bye. Thank you. Bye.